In this video, we will discuss about some of the patient samples RNA-seq analysis, and the focus will be on the patients that have this PPP2R1A in wild type conditions and the patients that have this PPP2R1A in the mutant conditions. And we will also check the effect of this mutant on the after or on this ICB treatment, that is immune checkpoint blockade treatment. So in total they have analyzed around 29 patients, and what they are able to see is that in the wild type case they can see some of the pathways that are upregulated, that are shown on the left hand side, that can be seen here as this cross signaling, apical junctions, ostrogen response late. However, the mutant PPP2R1A shows some of the upregulations of the immune response pathways such as iron gamma response, and then there's TNF signaling via NF kappa beta. And this is before the ICB treatment. And they also analyzed the RNA-seq analysis of the patients that have undergone this ICB treatment, and what they are able to find out is that the patients that have undergone ICB treatment has more significant upregulations in the immune response, such as IL-2 stat signaling, IFN alpha response, IFN elevated IFN gamma response, allograft in the rejections, and this inflammatory response. So this all in together indicates that in the mutant conditions of this PPP2R1A, the ICB treatment efficiencies increases, and it will enhance the immune response of the host or the patients. Thereafter, they did the longitudinal analysis of different immune cells. First, in the panel C, they have shown the result of about CD8 plus T cells, and we can see that in the mutant conditions, the enhancement is statistically significant. But in the case of this wild type PPP2R1A conditions, the enhancement on ICB treatment is not statistically significant. P value is around 0.1563. And when they check about these NK activated cells, they can see that it is also enhanced in the case of PPP2R1A mutant conditions. But in the case of wild type conditions, the values are not statistically different. And for the resting T cells, we can see that in the resting C cells, the difference is in both the cases is not that much statistically significant. However, it seems like that the resting NK cells is slightly higher in the case of the mutant conditions before the ICB treatment. And for the total NK cells is shown in the panel F. We can see that total NK cells are also almost similar. The difference is not statistically significant for both the cases, that is whether the PPP2R1A present in the wild type conditions or in the mutant conditions. The total NK cells remain same. However, just the activated NK cells increases on ICB treatment in the mutant conditions. Then they also check the T cell receptors and the B cell receptors. In terms of the T cell receptors, we can see that the enhancement on ICB treatment in the mutant cases, however, the p value is 0.06, but for the case of the wild type PPP2R1A, the p value is around 0.57. So there is increase in the mutant conditions to some extent. And when they check it for the B cell receptors, the increase is statistically significant. That can be seen here. The p value is around 0.03. It is less than 0.05 for the mutant case, that is, and PPP2R1A is mutant. However, when it is present in the wild type conditions, there is no statistically difference, as the p value is around 0.9375. So this in all together indicates that whenever this PPP2R1A present in the mutant conditions in the patients, it will enhance the ICB treatment effect in terms of increasing the immune response of the patient. So that's what they have presented. in this figure. So if you need to understand more about the experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.